Cell transport. When thinking about how cells move material in and out, it can be helpful to think of a cell as a nation. The boundaries of a nation are its borders, and nearly every country tries to regulate and control the goods that move across the border. Cells have their own borders, which separate the cell from its surrounding and also determine what comes in and what goes out. How can a cell be separate from its environment and determine who or what is allowed to enter and leave? What is passive transport? The movement of materials across the cell membrane without using cellular energy is called passive transport. Every living cell exists in a liquid environment. One of the most important functions of a cell membrane is to keep the cell's internal condition relatively constant. It does this by regulating movement of molecules from one side of the membrane to the other side. The cytoplasm of a cell is a solution of many different substances dissolved in water. In any solution, solute particles tend to move from one area where they are more concentrated to an area where they are less concentrated. The process by which the particles move from one area of high concentration to an area of low concentration is known as diffusion. Diffusion is the driving force behind the movement of many substances across the cell membrane. Suppose a substance is present in unequal concentration on either side of the cell membrane, higher concentration, lower. If the substance can cross the cell membrane, its particles will tend to move toward the area where it is less concentrated until it is evenly distributed. One comes in, some they might go out, but then more come in. Diffusion causes a net movement of solute particles from the side of the membrane with the higher solute concentration to the side with the lower solute concentration. At that point, the concentration of the substance on both sides of the cell membrane is the same, and equilibrium is reached. As you can see, they still cross, but they cross evenly. And that's equilibrium. No net change in solute concentration on either side. Even when equilibrium is reached, particles of solution will continue to move across the membrane in both directions. Because almost equal numbers of particles move in each direction, there's no net change in the concentration on either side. Travels here, travels there, travels here, travels there, equally. Diffusion depends on random particle movements. Substances diffuse across membranes without requiring the cell to use additional energy. The movement of material across the cell membrane without using cellular energy is called passive transport. Now we will cover transport that is aided. Cell membranes have proteins that act as carriers or channels, making it easier for certain molecules to cross. Molecules that cannot directly diffuse across the membrane pass through special protein channels in a process called facilitated diffusion. Hundreds of different proteins have been found that allow particular substances to cross cell membranes. The movement of molecules by facilitated diffusion does not require any additional use of the cell's energy. Osmosis is an example of facilitated diffusion. The inside of a cell's lipid layer is hydrophobic or water hating. Because of this, water molecules have a tough time passing through the cell membrane. Many cells contain water channels, the proteins called aquaporins, that allow water to pass through them. Without aquaporins, water would diffuse in and out of cells very slowly. The movement of water through cell membranes by facilitated diffusion is an extremely important biological process, the process of osmosis. So the cell membrane will block because the hydrophobic tails, but this aquaporin will allow the water to flow through here, either direction. Osmosis is the diffusion of water through a selectively permeable membrane. Osmosis involves the movement of water molecules from one area of higher concentration to an area of lower concentration. In an experiment set below, the barrier is permeable to water, but not to sugar. This means that water molecules can pass through the barrier, but the solute, sugar, cannot. So the membrane is here, set in place, set with low concentration of sugar and high concentration of sugar. So after a time, the water will cross the membrane to make equal concentrations of solute. So the water will naturally go through the aquaporins to make the concentration of sugar on both sides equal or close to equal, even to the point of going against gravity flow. This will have more gravity pressure, but the solute high concentration on this side will pull in more water to make this more balanced. There is more sugar molecules on the right-hand side of the barrier than on the left-hand side. Therefore, the concentration of water is lower on the right. So low concentration of sugar, higher concentration. There's a net movement of water into the compartment containing the concentrated sugar solution. Water will tend to move across the barrier until equilibrium is reached. At that point, concentration of water and sugar will be the same on both sides. Same concentration of sugar. Notice osmosis will even work against gravity, and there will be more pressure over here, but because they're equal concentration, it's balanced. When the concentration is the same on both sides, the two solutions will be isotonic, which means same strength. So these are now isotonic, the same concentration. The more concentrated sugar solution at the start of the experiment was hypertonic, above strength on this side, compared to a weak dilute sugar solution on the left-hand side. The dilute sugar solution was hypotonic, or below strength. For organisms to survive, they must have a way to balance the intake and loss of water. The net movement of water out or into a cell exerts force known as osmotic pressure. Because the cell is filled with salts, sugars, proteins, and other molecules, it is almost always hypertonic to fresh water. As a result, water tends to move quickly into a cell surrounded by fresh water, causing it to swell. Eventually, the cell would burst if left in fresh water. In hypertonic solution, water rushes out of the cell, causing animal cells to shrink and plant cell vacuoles to collapse. The solution has a higher solute concentration than the cell. A net movement of water molecules out of the cell causes it to shrink. Some have poured salt on a slug, and that's what happens here. The salt concentration on the outside of the slug will pull the moisture out of the slug. But in normal situation, the cells are bathed in fluids such as blood that are isotonic and have concentration of dissolved materials roughly the same as those of the cell. Cells placed in isotonic solution neither gain nor lose water. The concentrations of the solutes inside and outside are the same. Water molecules move equally in both directions, in both plants and animals. In plants, the movement of water into a cell causes the central vacuole to swell, pushing cell contents out against the cell wall. So gaining water here will reinforce the cell wall. Since most cells in large organisms do not contact fresh water, they are not in danger of bursting. Some cells, such as the eggs laid by fish and frogs, must come into contact with fresh water. These type of cells tend to lack water channels. As a result, water moves into them so slowly that osmotic pressure does not become a problem. Notice that a plant cell holds its shape in hypotonic solution, while the animal Notice that a plant cell holds its shape in hypotonic solution, while the animal red blood cell does not. Other cells, including those of plants and bacteria that come in contact with fresh water, are surrounded by tough cell walls that prevent cells from expanding, even under tremendous osmotic pressure. However, the increased osmotic pressure makes such cells extremely vulnerable to injuries to the cell wall. What is active transport? The movement of material against a concentration difference is known as active transport. Active transport requires energy. Cells sometimes must move materials against a concentration difference. A considerable portion of energy used by cells in their daily activities is devoted to providing the energy to keep this form of active transport working. The use of energy in these systems enables cells to concentrate substances in a particular location, even when the forces of diffusion might tend to move these substances in the opposite direction. The active transport of small molecules or ions across the cell membrane is generally carried out by transport proteins or protein pumps that are found in the membrane itself. Many cells use such proteins to move calcium, potassium, and sodium ions across cell membranes. Changes in protein shape seem to play an important role in pumping process. So they use ATP to pull in a molecule from one side and pump it to the other side through this protein pump. Active transport of larger molecules and even clumps of material across the cell membrane is done by a process known as endocytosis and exocytosis, commonly known as bulk transport. The transport of these larger molecules often involves changes in the shape of the cell membrane. Endocytosis, the members form a pocket around the particle. The pocket then breaks loose from the outer portion of the cell membrane and forms a vesicle within the cytoplasm as it comes in. So it will grab around it and then break off and become a vesicle inside the cell. Exocytosis, the membrane of the vesicle surrounds the material, then fuses with the wall membrane. The contents are forced out of the cell when it opens. So this vesicle is joining against the wall, and then it wraps open here and the contents is pushed out. From exocytosis, many cells also release large amount of material from the cell, a process known as exocytosis, as we talked about. During exocytosis, the membrane of the vesicle or the vacuole surrounding the material fuses with the cell membrane, forcing the contents out of the cell. Endocytosis is the process of taking material into the cell by means of enfolding or pockets of cell membrane. The pockets that result breaks loose from the outer portion of the cell membrane and forms a vesicle or vacuole within the cytoplasm. Right here, we'll encapsulate this piece and make a vesicle. Large molecules, clumps of food, or even whole cells can be taken up by endocytosis. Two examples of endocytosis are phagocytosis and pinocytosis. In pinocytosis, cells take up liquid from the surrounding environment by forming tiny pockets along the cell membrane. The pockets fill with liquid and pinch off to form vesicles within the cell. These include cells of the liver, kidney, and skin, the epithelial cells. In phagocytosis, extensions of cytoplasm surround a particle and package it within a food vacuole. The cell then engulfs it. Amoebas use this method for taking in food. Engulfing material in this way requires a considerable amount of energy and therefore is a form of active transport. These include immune first responders like macrophages, white blood cells.